Hi, everyone, and thank you for joining us today. Welcome to today's Microscopy Masters Webinar with Dr. Daniel Rolfe from the Octopus Imaging Facility at STFC, focusing on automating FLImP, macromolecular structure fingerprints for improved cancer therapy. Before the webinar begins, we'd like to go over some quick house rules for the webinar. I'm sure you've all noticed by now, but you do have a webinar navigation panel on the side of your screen containing a few different sections. If you'd like to more clearly see one specific section, you can simply click the expand icon, which is located in the top right corner of each section. Today's webinar will last about 45 minutes, leaving approximately 15 minutes for Q&A at the end. You are welcome to submit any questions that you have throughout the webinar via the chat section located within your panel. However, we will only be answering your questions during the Q&A session. If we were unable to get to your questions, then we will email you answers separately via the email address in which you registered with. Within your navigation panel, you will also find a handout section, and here you can find our company brochure telling you a little bit more about ONI and our offerings, so please feel free to have a look there or on our website at oni.bio. Please note that today's webinar is being recorded, so within the next day, you will receive a thank you for attending email with a copy of today's webinar to refer back to, in addition to a video recording of today's webinar that will also be stored on the resources section of our website. Now I'd like to introduce you to our presenter for the day, Dr. Daniel Rolfe. So Daniel leads the data science and computing efforts at STFC's Octopus Imaging Facility with a particular focus on quantitative single molecule microscopy. And he has been doing this as part of a growing multidisciplinary team over the last 17 years. And something very interesting about Daniel, which we just discussed right before this webinar, is that he had a previous career as an astronomer and an astrophysicist, followed by a brief period developing protein crystallography analysis pipelines before being tempted into the world of single molecules. And with that, I will now pass the presentation over to Dan to begin the webinar.
Brilliant. Thank you, Carolyn. Thank you, ONI, for inviting me to talk about our work myself and doing with a large team over some time. So this is automating FLImP, macromolecular structure fingerprints for improved cancer therapy. And as Carolyn says, I come from the data analysis, data science, and I spent a long time in astronomy. And as an astronomer, people assume that I could point them to lovely constellations and produce brilliant pictures of galaxies, but actually I spent 4 years studying 2 dots. So you'll see a talk today where you'll see the results of studying millions and millions of dots in great detail. So it's a step up for me. So without delay then, so I work for Octopus. So it's got a nice acronym that trips off the tongue. It stands for Optics Clustered to Output Unique Solutions. So we are part of the UK Research and Innovation Science and Technology Facilities Council's Central LACE facility. So it's a national, a government national facility and a national lab. We're based on Harwell campus, which is about 16 miles to the south of Oxford. So Oxford's up here somewhere, I think, or up there, off on the horizon. So we're spread across this campus. So this is the Diamond Light Source, which is our national synchrotron facility. And when we're sort of spread in 2 locations across the campus, and the facility is sort of divided in terms of techniques into two sort of different groupings, all with lasers at the heart of some of the methodology. But we have the high power lasers part of the department, so 2 facilities, Astra, Gemini and Vulcan, but they're about super high power, some of the high power lasers in the world to produce ultra intense lasers for producing extreme conditions and extreme condition science and laboratory astrophysics. So super high power lasers there. And then I work in the Lasers for Science facility, which contains 3 facilities in itself. So we have Artemis and Ultra, which are ultra fast spectroscopy facilities, so looking at chemistry, ultra fast dynamics, from infrared through to soft X-rays. across this facility. And then I say I'm part of Octopus, which is an imaging and laser facility. So in the Lasers for Science, it's very much about what we use the lasers themselves for, rather than it being super powerful in themselves. So there's also a facility that the Central Laser Facility is just developing at the moment called the Extreme Photonics Application Centre. This is a new £80 million centre for laser-driven sources. It's actually developing a new type of accelerator, so using a powerful laser as a plasma accelerator, firing that at targets to do a whole new range of science. So that's going to be operational in a couple of years' time. So the Central Laser Facility is a wide variety of facilities with lasers at the heart. But so a bit more about Octopus. So we're based here in this little glowing part here. So we're right next to the diamond light source. This is the research complex at Harwell, which is a multidisciplinary research institute on the site, bringing together scientists from both physical and life sciences to take advantage of and collaborate using the various facilities on site. So Octopus then is an imaging facility, imaging across scales. We have a wide variety of techniques in Octopus. So from FLImP, microscopy, single particle tracking, we have laser tweezers, super resolution methods, so SIMS, STORM, so on. And we also now have pro electron microscopy, so FibSem microscopes, and we have light sheets. We have the first min-flux super resolution microscope in the UK, and also what I'll be talking about more is FLImP, but the key, it's about sort of imaging, and particularly molecular imaging, across a wide range of scales. So in what I'll be talking about, particularly sort of trying to understand molecular structure function relationships on sort of scales of 1 to 10 nanometers, a few tens of nanometers, but going right up to whole organisms. So it's all about being able to image across the scales. We provide the cluster of microscopes. They're both commercial instruments and in-house developed instruments, and we provide those along with expert end-to-end multidisciplinary support. So you get access to help from people like myself, who used to be astronomers, mathematicians, through to biochemists, biologists, and microscopists as well. And so to help people with their science, we're a national facility with peer-reviewed funded access. So when people actually apply, if they get access to us, then we pay for then essentially everything's paid for: their travel, their accommodation, and consumables and so on. And developments of our capabilities, they're funded both through external grants and by funding from within the organisation. So a lot of what I'll be talking about will be techniques we actually developed to understand a particular system, and so a lot of that work was actually developed by external grants, and then we translated those methods into the facility and are now making them available to others. So the subject that's driven a lot of the single molecule technique development and motivated a lot of that in Octopus, and that's particularly driven by Marissa Martin-Fernandez, who is the head of the group, head of the Octopus facility, and she's really been the driving force between the EGFR research in the group over the last, well, more than the 17 years since I joined. She tempted me into joining her in this project. So this is studying the epidermal growth factor receptor. So it's a membrane protein receptor. So it's effectively, so remember, this is an astronomer talking to you, explaining this. So it's essentially a molecule in the membrane of the cell. It effectively acts like a switch. So it responds to a molecule's binding from the outside. So effectively a chemical stimulus somehow or other, and this we'll go into, it changes its structure or interacts with other molecules, leading to change of its structure within the cell, which ultimately propagates a cascade of interactions within the cell to the nucleus. And in the case of the epidermal growth factor receptor, that can lead to cell death, cell migration, cell growth, adhesion, differentiation. When that goes wrong, a key problem when that goes wrong is you get cancer. So if a cell doesn't die when it should, goes where it shouldn't, grows out of control, that's cancer. So epidermal growth factor is heavily implicated in a wide variety of cancers. For example, breast cancer. It's a key target for cancer drugs. Things like Herceptin are all about disrupting how it works. So understanding how EGFR works has been what's driven research and what tempted me to join this and the development of single molecule methods to study this. So it's been understood for a long time that the mechanism involves, I say, so the earliest model for it, and one of the sort of lasting models, is actually something called ligand-induced dimerization. So this is where EGF, which is the molecule which binds to the EGF and actually causes it to do things. So it binds. The idea is that then it binds to monomers. They end up coming together and producing dimers. That changes their conformation externally and then internally, and that leads to then them propagating the sequence of cascade of interactions inside and actually doing something. It's become clear over the years since then that it's actually more complicated than this. So ligand-induced dimerization is part of the story, but it's actually definitely involves larger order oligomers, possibly not always in the presence of ligands. Some of the earlier work we've done with FLImP and other studies has actually fed into some of these discoveries. So there's a lot more to understand, and that's critically important in understanding and treating cancers. So the simplest understanding of it led people to try and interfere with this process, and that things like herceptin. So that means they actually are able to start tackling and improving people's chances with cancers. But you also get nasty side effects because it's a bit of a sledgehammer to crack a nut. It's far more complicated, and so we're turning off things we want to and things we don't. So understanding it better is really critical to actually improving people's chances with cancer survival. So single molecule microscopy. So why do we use single molecule microscopy? So as I say, key developments in Octopus in the single molecule side of things have been driven in particular by, some of them have been driven by the EGFR study and the motivation to do that. So TIRF single molecule microscopy, so that's where you use total internal reflection, so you end up illuminating a very thin layer on the surface of your sample. So if that's just essentially the molecules on the surface of your cell in the membrane, that reduces the emission from the background, and then you can get images like this. So this is actually the same field of view on a TIRF microscope. So it's the same field of view in both channels. These black dots correspond to one or a very small number of fluorescently labeled EGFR molecules, and we can then run them through our analysis, and we see they're moving around, so we can study their dynamics, but we can also look at when they come together by seeing when they're in the same place in the two channels. You might, for example, be labeling different parts of the molecules in the two different channels with two different fluorophores, but you see how you can get very rich asynchronous events to actually understand exactly the dynamics and the interactions between the things you're labeling, and that's why single molecule microscopy is something that was really something they were trying to apply to understand the epidermal growth factor receptor interactions. So when we sort of started this project, or before I even joined, Marissa and a few colleagues had sort of built their first single molecule TIRF microscope, and they were starting to get data like this. And in fact, it was actually then that they were seeing that these things looked like star fields in many cases and looked like galaxies, and they didn't know how to analyze them, and they tried to find an astronomer. And that's how I was tempted in this part-time job for initially helping on the side, and then 18 months has now turned into 17 years so far. So certainly sucked me in. So understand these single molecule images is critical, but actually how do we actually measure what we're interested in? So we're trying to measure the interactions. So this is again the original sort of the traditional dimer model. So here we have the fluorescent dyes attached to our EGF ligands. So when we're trying to understand and measure these structures and determine their interactions, we need to be able to image and measure things on the scale of about 12 nanometers. So these dyes are basically separated by about 12 nanometers when they're attached here. So at the time, sort of 15 to sort of 17 years ago, when we were looking at this, the super resolution techniques that were starting to become available, like Storm and so on, they sort of could deal in the range of several tens of nanometers upwards. An alternative method, which was fluorescence resonance energy transfer, which is a dipole-dipole interaction, which basically allows you to determine with two-color microscopy, single microscopy, determine the separation of labeled fluorophores on the scale of about 0 to 10 nanometers. Unfortunately, what we needed to do to understand these processes was look in that gap between about 10 and say 40, 50 nanometers. So there was a gap there that wasn't really easily accessible and measurable with the existing techniques. So this is what motivated us to actually develop the FLImP technique. So FLImP, so we have another beautiful acronym here. So FLImP stands for Fluorophore Localization Imaging with Photobleaching. So in this technique, so it's a single molecule TIRF technique, and it starts with tracking essentially. So we fix our samples before imaging, and then we exploit photobleaching to measure the subdiffraction limit separations of the labeled receptors. So here, these are the two fluorophores separated by around 10 or 12 nanometers. However, this is the point spread function size of an optical microscope, sort of 400, 500 nanometers. So we're never going to actually look at that and determine the separation or localize these two molecules separately. However, if we perform single molecule tracking on this spot. So we followed it through time. So this is now the intensity as a function of time of this single molecule spot, one dot in an image. And we can, and for examples where we have, say, we start with 2 fluorophores first, and then 1 photobleaches, and then followed by the next one. So we have this sort of textbook photobleaching example. In this period where only one of the fluorophores is on, so here, we just have the profile of one fluorophore, and so we can actually do, essentially, we'll just do fit a Gaussian, say, to that profile. So fit the profile and just essentially limited by the number of the number of photons, or set by the number of photons, we can actually very precisely localize this molecule on its own. So once we precisely localize this one, then we've also got a localization of the pair together. So this one plus one more, but knowing the localization precisely of the first one, we can then infer where the other one was by measuring them together. So that's basically the principle of the information we're using. And what we actually do is, rather than separately localizing them, we fit a model to the region of interest of the profile changing through time. So we know from the intensity trace when it turns off, so when the photo bleaching steps are, we fit a model essentially to the region of interest throughout the whole time period of 2 then 1 molecules, and then we can actually jointly fit the locations and localize the 2 molecules jointly. We actually use bootstrap resampling of the pixels through time that we have in our data to actually empirically infer the uncertainties in our measurement, so we can actually then localize our 2 molecules to a small number of nanometers localization precision, and we can measure the separation between the molecules present, and from the bootstrapping we can actually get an uncertainty, so we actually essentially get a probability distribution for the separation like we get here, and with errors, certainly in this is sort of data when we first developed this method, sort of around sort of 5-7 nanometer sort of uncertainty, and we have to correct for the drift in the sample, which is an important part of the process. But that's the key principle of the method, is fitting the profiles of photo bleachers and the change between 1 and 2 molecules being present or visible fluorescing. So if we now imagine we have many such spots, possibly from multiple data sets, so we find all the spots in our data sets which have those nice photo bleaching steps so that we can apply this method to them, and we get many of these probability distributions, so essentially separation measurement with uncertainty. We have many of those and we pull them, and then if we imagine, so we fixed our samples before we imaged them, and then on the assumption that we think is reasonable that there's a fixed number of discrete structures that our molecules are forming in the membrane once we fix them. Each of these measurements is essentially one separation, is essentially a randomly chosen separation between any of the possible labeled locations within our structures. So if we measure many such separations and pull them, what we'd expect is to see components peaks with essentially a fingerprint with peaks corresponding to the signature separations between our molecules, between the labeled locations. And so we can then infer from that information about the structures present, and particularly if we then combine this with PDB models, molecular dynamic simulations, we can actually then start using these fingerprints, as we call them, these FLImP fingerprints, to actually infer and get detailed understandings of structural changes, and we can compare between different conditions and so on. So here, and in this case, so this grey histogram here is essentially a sum of all these probability distributions, which we can then fit as a sum of Rice distributions. So that's basically just the probability distribution you'd get for the separation between 2 Gaussian localizations. So we can fit that with uncertainties, decompose it into a model saying what are the discrete separations that fit this data set. And so our first sort of 2 major sort of papers where we're applying FLImP from 2016 and, no, when is it? Come down here, I'm losing track of time now. From 2016, 2019, we applied this method, and essentially we were able to already gain new insights into epidermal growth factor signaling from our sort of 5 nanometer resolution snapshots of the signatures, and we started getting new insights, in particular the presence, demonstrating the presence and the role of the larger EGFR oligomers in the EGFR function, and also the existence of preformed EGFR oligomers, so the presence of oligomers even without ligand. So this was really our first work with FLImP. So we developed the method and we applied it, but this was really painful to do. So although the method is in principle very simple, there's a lot of manual work to actually apply it, to get enough measurements, to get useful FLImP signatures to answer questions and to do science. So particular work involved is, well, I'll go through that now. So what we had then was a vision to actually automate this so that we could actually do it much less painfully. so much more easily and make it much more practical to apply in reality. And we actually had a vision that we might actually then be able to apply this in the clinic. So we imagine, so the original idea was we thought it would blend itself well to non-small cell lung cancer as an example. So where you would have a patient biopsy, a liquid biopsy, you put it on the slide, you put it onto some, if we could imagine there might be an automated single molecule microscope somewhere, if there was such a thing, then we would put it on there and it would automatically run through all the analysis to the point of producing our FLImP signature. And so then we're automatically providing this detailed molecular signature of the separation distribution that we can then start understanding things about the data and about the molecules. But if that was then a patient's biopsy, you can see how you're getting a detailed molecular fingerprint of what's going on at the molecular level in that patient. You can compare and contrast those between patients at different stages of their treatment, compare it with studies of cell lines in the lab, for example, and start making patient-specific predictions about which particular treatments, for example, might work well for particular patients. So that was the vision we had, and we got funding from STFC from a particular funding round for the translation and commercialization of techniques in areas such as healthcare. And so we got money to fund essentially an automation project to do that. And so what was actually needed then, and I'll focus on really this blue box here. So this is about automation of the method. So what's actually needed to automate? I said it's very painful. So it's actually the identifying the regions of interest in your samples to actually acquire movies for, which we need many of those to build up our data sets, identifying the individual tracks that are sufficient quality to analyze, running through the analysis to produce the fingerprints. So that's what was needed to be done. So the vision was, let's do that, let's try and do that, and if we can automate it, then the full automation will increase throughput right from acquisition to output. It'll increase our efficiency and impact in lab research, the sort of thing we've already published, and also make potential for applying this as an automated structural fingerprint for personalized cancer care. So we had the funding, and a colleague called Ben Davis joined us in 2019 to start on that. So I'll now go through a few of the steps we took there. So as I said, we need to imagine some nice automated microscopes. So we started with, we've developed FLImP on our mostly manual Zeiss microscope using in-house data acquisition software written in LabVIEW. Sorry, data acquisition software written in LabVIEW. Pretty much everything you want to do involves twiddling a knob, finding a field of view by hand, manually doing almost everything. And so what we needed was to try and find a microscope that we could actually take all that manual process out. So anything we can imagine a code being able to work out how to do, we wanted the code to make it happen. And we came across this thing here. And so we ended up trying this out, and colleague Sarah, she went and she tried out, she went and visited ONI, so it was very convenient that we happened to be down the road from Oxford, which is where ONI were based. And we tried out some samples on a Nanoimager to see if we could do FLImP, get the sort of quality data on there, which we could. So we, as part of our grant, then we bought one of these. So the advantage for us, I mean, it's a lovely, obviously a lovely desktop compact instrument, which is great for putting in the clinic, but most critical for us in this project was just the ability to automate. So we wanted everything the microscope could do to be software controllable, and ideally, as it was, and as it is, Python scriptable. So so basically, you know, a super compact TIRF capable single molecule microscope that we could fully control with software. So we bought one of these and implemented our data acquisition pipeline then within that. So what we actually needed to do was to automate the region of interest identification, so finding suitable fields of view within our large slides to take FLImP movies. So we did that in 2 steps. So first of all, we stained the cells with Hooks, which is a nuclear stain, nucleable, to identify basically where the living cells were, so we could home in on regions that had basically cells in them in one channel. And once we identified regions of interest where sufficient areas, so we did simple OTSU thresholding to actually essentially to split that into foreground and background. And if a sufficient region within a field of view was to have cell in, then we thought that was a starting point for a good field of view. We would then look in the single molecule fluorescence channel, either FLImP channel, and use triangle thresholding to identify how much of the region of interest there was actually like it was well labeled for the FLImP. And basically we would then settle on regions where a sufficient proportion of the field of view was both had live cell and had single molecule labeling. We would then acquire a movie of that. So that already transformed the acquisition, because previously there was lots of manuals hunting around for regions to take movies of. So Ben also did try deep learning, so he tried to apply UNet to this problem, and unsurprisingly it worked very well. It lends itself, bread and butter problem for techniques like that. However, we stuck with the much more simple deterministic approaches because it's sort of still an issue to actually get deep learning methods through to be approved in medical devices. So given that a simpler method actually worked, we stuck with that, but we've shown that it's something that can be done very well with deep learning as well. So, but we stuck with our simple method for that because it worked well. So what we also need to do was autofocus refinement. So this is finding the initial focus on our single molecules. So what was done then was to scan through, to basically take a Z stack of images through the point spread function. So obviously where it's sharpest is where it's best in focus, and then a Gaussian kernel with the PSF, about the PSF of the instrument was then cross-correlated with the images, and basically where the correlation was sharpest, so where the point spread function was sharpest, that was when we were in focus. So that would enable us to identify a good focus, and then we can switch to the Nanoimager to maintain very effectively the focus with this autofocus from that point once we'd found the optimum focus for our experiment. So having got our automated microscope, having found our ROIs, having focused well on our simple molecules, we then acquire a movie, so it would be about 1200 frames at 20 millisecond exposures. So this automated acquisition enabled us to go from 50 videos a day to the order 100 an hour. So that already transformed the rate at which we were able to acquire data. And that was really something which we needed. With both the algorithms and the scriptable single molecule microscope were essential for that. So also part of this, just improving the user experience. So our ideal user interface for this microscope, for this particular workflow, is one button that says go. So at the moment, there's a few more things we have to control, but essentially we minimize that by providing a sort of a limited set of inputs to try and guide the user away from interfering with messing with parameters they don't need to. So we make it as simple as possible. And then also sort of real-time performance monitoring, so a wide variety of diagnostics running as the instrument runs, because it runs sort of essentially, you put a sample on and then it runs for potentially 24 hours acquiring data, but it will tell us which, how it's covered the fields of view across the slide as it's gradually working its way across acquiring data, and a variety of diagnostics, so to give us feedback in terms of the performance and quality metrics as it's acquiring the data. So that's the acquisition side of things. So then what we also needed to do was automate the analysis pipeline. So actually essentially getting the FLImP fingerprints from our data. So one step of that, which was previously very manual, was the drift correction. So this is correcting for sample drift. So in the first versions of FLImP, we essentially would average the motion of all our single molecules in our field of view, fit a polynomial to them, and there was manual oversight to basically identify where we think that would work well enough, acceptable regions for the nice smooth fit. So that already made it manual and slow. So what we wanted to do was automate that. So what we actually then do is put separate bead fiducials into our samples, which are nice and bright and on for the whole period of the data, and then we can do automate. Because we've already tracked them, because we do single molecule tracking the first part of our analysis, we can from those tracks identify which ones were beads. We can actually just remove outlier steps in their motion and do cross-validation between different partitions of the beads to basically identify the good quality beads from which we then identify the drift, and that enables us to get the drift essentially the drift down to sort of uncertainties of about 1 nanometer. So this is sort of X and Y components of drift in green and the residuals in red, just one example. So key to that then, so the result of that was both a better drift calculation, but also an automatic drift calculation. So the other key part of the analysis automation is track selection. So as I showed you, basically we have to pick out the tracks in our data that look like this. So nice single molecule photo bleaching steps. There are various other requirements for the analysis, such as they're not sort of blended with other features nearby, which will interfere with the fit, for example. And so previously, in the first version of FLImP, this was done with sort of manual inspection. So there'd be some filters in our single molecule analysis software to help home in on the right tracks. But ultimately, there was Sarah, who's a key member of the FLImP team, spent many days and hours manually going through to identify the tracks that were actually suitable for FLImP. So this is not choosing ones that give the answer we want. This is just choosing the ones that are suitable for a measurement. So that fit the assumptions of the photo bleaching FLImP process. So what we wanted to do then was go to a fully automatic process for this, and we actually came up with essentially a fairly straightforward set of explicit criteria. You know, the principle of the problem is relatively straightforward, but there's a whole load of assumptions it makes that you want to test for in your data. And we actually found that a set of very explicit deterministic questions worked. So we actually like play a game of guess who. So essentially the single molecule tracks in each data set. We asked lots of questions of their position and their various properties through time, and gradually whittle down from perhaps 10,000 or tracks in a data set down to the small number that are actually potentially going to yield good measurements for FLImP. And so by applying that, we went from track selection of about 100 tracks per day and lots of hair tearing out from Sarah and other colleagues to millions an hour. So that's basically the key steps we had to do to analyze, to automate our analysis. So then we had to deploy this. It's quite compute intensive. So we had to deploy the whole processing pipeline then on a compute cluster on our STFC cloud, which is funded by IRIS, which is essentially pooling together of all the STFC facilities to bid for and sort of coordinate their high performance computing. And from that we were able to secure quite a significant amount of compute resource to help with this project, without which we wouldn't be able to do it. And colleagues in the scientific computing department here who actually operated a lot of that for us. So that was essential for us to do this. So we have sort of 50 CPU machines and 50 graphical processing machines, and also a sort of a distributed file store with over the last few years acquired about 800 terabytes of data from this one little Nanoimager. So lots of computers, perhaps more than this, and lots of storage. So perhaps a few more computers and perhaps some slightly more up to date, the ones we actually use. But yes, lots of compute to deploy this pipeline. So the progress we've made then, so we call this FLImP next generation. So this is the automated FLImP. So from this automation of the acquisition through the algorithms applied on the Nanoimager automating scriptable instrument, and we reduced 1 month of acquisition and analysis now down to 2 or 3 days essentially. The better drift correction and the fact that we have just far more data means we have far more high resolution data. So this little bar chart here is actually, sorry, it's a bit small, but this is May 2019. So this is essentially I think the number of FLImP videos we require per day. And this is in May 2019. So this is just around the time Ben joined us to automate it. We sort of successfully automated around here. Something happened around March 2020, which is why our numbers went down again. But you'll suddenly see that by summer 2020, the actual data acquisition, it was increasing by a factor of perhaps 50 to 100 over a few months, and it maintained steadily at that rate. And bearing in mind that the vast majority of our team were working from home from about here, in case of the data analysis team, we were working home from here to probably about this plot here. We were only able to do the work I'm talking about, that I'll talk about next in terms of applying FLImP, because fortuitously we had actually automated FLImP a few months before the pandemic. So if it hadn't been for that, none of the work I'm now going to talk about would have happened, because we wouldn't have automated it. We were able to do this work with our Nanoimager automated analysis just with someone who was able to come into the lab, because a small number of people could come in, and who was actually able to put the samples on the microscope, and everything else was basically happening remotely. So this enabled us to acquire far more data, enabled us to actually take data at all during the pandemic. We also improved our decomposition algorithm, which was something that the pandemic gave me and Ben a lot of time to think about, that because we didn't have certain other things we would be doing while we were stuck at home. So we had a bit more time to actually improve on some of this. So the algorithm I talked about, which takes the many FLImP measurements and then interprets them in terms of a fixed number of discrete separations. So it essentially finds out what the separations are in the FLImP signature. I actually developed a much more, an improved algorithm which is more optimal. So a more optimal Bayesian algorithm that, rather than fitting the sum of the individual measurement histogram, essentially posteriorly, actually takes them all separately, but and jointly analyzes them with a Bayesian approach, still with the same model. So essentially asking, if all these data come from a discrete set of separations, in reality, what are the separations and how many are there? But doing it optimally, considering all the separations separately, it samples from the assignment. So thinking each individual measurement in reality will, in the model, corresponds to one of the separations we're going to end up fitting. So you have to explore all those possible ambiguities in assignment. You also have to account for the fact that there are spurious measurements, so some measurements would be wrong. So basically it's a Markov chain Monte Carlo sort of Metropolis Hastings algorithm that actually explores all those possibilities to decompose, get lots of complicated plots here, But the result was that we could end up getting down to, with both our improved data and this more optimal analysis, down to 1 nanometer precision in our decompositions to actually interpret our FLImP signatures. So this is really now what we call the FLImP next generation. So this is what we've been running, and essentially we're able to run 24/7 during, well, for a lot of the lockdown, or perhaps 6, because it depended on somebody able to go in and put samples on the microscope still. And so essentially we fully automated this blue box from our vision that we got for our grant. So basically fully automated the acquisition through to producing the FLImP signatures, and indeed actually the ability to decompose them. And so the human effort there after sample preparation was minimized. It's compute and data intensive, which is probably out of date by now, but a couple of million jobs on our cluster, and I'd say 700-800 terabytes of data so far. There's a lot of potential for more compute and data efficiency improvements, and so therefore also cost and energy efficiency improvements, which we're looking at now. Now, the other part of our vision we didn't make so much progress with in terms of the application in the clinic, and that's, I think, the thing that we weren't really able to get around during a pandemic. So obviously with a global pandemic of a lung disease, our clinical collaborators who were in respiratory medicine were somewhat preoccupied. So we're looking now at how we can continue with that. But the bit we were able to do, the acquisition of the automation of the acquisition and analysis, essentially we think we were very successful with that. And then this then just demonstrates what the automation and remote access enable us to do. So Ben, who I say did a lot of the automation of this on the grant, so he, like me, was stuck at home for the best part of 18 months, but with Sarah putting samples on, preparing samples on the microscope, was able to come in. This is Ben having a walk through the park and keeping an eye on how our FLImP acquisition was actually going on the ONI from his mobile phone. So it sort of illustrates that, we couldn't have done this work without the automation, particularly given what happened around the world. So I'll now move on to what this has actually enabled us to do. So going back to what motivated all of our single molecule sort of developments, and particular FLImP, and what we actually then have done and we're doing basically through the pandemic as well, was a FLImP-enabled study, and also a study enabled by the automated FLImP or ligand-free EGFR oligomer assembly, and particular focus on certain lung cancer mutations. So it was essentially a study of a wide variety of mutations and treatments. So it's basically in cells, it was CHO cells in the lab, Chinese hamster ovaries, but studying wide variety of interactions in EGFR, but taking FLImP measurements from them. But there's essentially a wide variety of interactions and mutations which are known to affect these interactions. Some of these interactions are things we actually ourselves identified or proposed in some of our earlier papers, but we essentially did a very complicated large study that was only possible with the scaling up we got from and the improved quality we got from the automated FLImP. So we labeled, these were all ectodomain labeling, so we either labeled with an anti-EGFR antibody. So this is all essentially ligand-free. So this is ligand-free oligomerization. So we either were labeling with anti-EGFR antibody with CF640R as the fluorophore, or in some cases it was EGFR, but we was in a couple of samples, but things were fixed before probing with EGF. So the idea that basically there shouldn't be any activation, but the vast majority of the data was actually, I say, antibody labeling. So that means it binds in the same place, but doesn't activate or change. Just doesn't do the same as the EGF does. So we basically did a huge study, taking lots and lots of FLImP data, running it through our automated pipeline, and then decomposing our FLImP signatures. One thing this enabled us to do then was to automatically compare and contrast these. So each of these decomposition, essentially this is just the probable distribution of the separation after decomposition with the Bayesian fit, and we could actually compare them all to one another we use something called the Wasserstein metric, which essentially is a score for how similar the different distributions are. But when they're not the same, it actually scores them worse depending on how far you would have to move probability basically to put it in the same place. It's like an Earth mover distance. But basically it's a measure of, are the distributions the same, and if not, how different are they? That sort of lends itself well to this problem. So we could use that to compare them all with one another and then put it into a dimensionality reduction algorithm. It's essentially a clustering to produce things like this, where essentially each of these colored blobs is counting for uncertainties that we actually obtain from bootstrapping the data sets as well. Essentially it tells you basically it puts into 2 dimensions clusters of locating the different FLImP signatures, so the information telling us about molecular structure. So things that are close together are the same, and things that are further apart are different. So it lets us start seeing which things, without actually asking questions separately of all of them, are similar or not. So that was already quite a nice automated way of starting to see which things are similar to one another. And that's a step towards some of the stuff we would like to go back to in terms of automated ways of exploiting these, for example, in the clinic, to compare and contrast patients, for example, so you can actually start making predictions. So that's sort of a nice step towards that. We also then used it in our paper, but where the real hard work in the paper came in really trying to understand what these measurements are telling us. So we're doing the decompositions, identifying the peaks in them, for example. Here's the clustering we did, but lots of work comparing, contrasting, and critically using molecular dynamics simulations. So we had some great collaborators in Geneva who did vast numbers of detailed molecular dynamics simulations of what we're looking at, and there's lots of interplay backwards and forwards, and trying to understand in the context of all the different mutations and knowing which interfaces they interfere with, and how we actually see components appearing, disappearing in the structures from the FLImP distributions, and seeing when you know, in some cases you treat with a particular drug and then you find out that that does the same as a particular mutation that's known to affect an interface. So lots of reasoning. My head was spinning. Marissa was scribbling and plotting away with her plots to understand this. But the result of all this vast amount of work that was depended on this sort of new sort of extra high resolution FLImP automation and particular molecular dynamic simulations, and what goes on in Marissa's head was this model for ligand-free oligomerization. So this is like a top-down view of the oligomers, and particularly what happened is we identified 2 previously orphan interfaces that interfaces which seem to be important after all. So this came entirely from our what we call the 1D FLImP, so just measuring separations. So this was a key step, but what we then did next to validate it was to use what we call 2D FLImP. So as I showed you, the basic FLImP method, you have 2 photobleaching steps, so you go from 1 to 2 molecules, and you can then further essentially localize the 2 molecules. If you have 3 steps, you can actually localize a triangle. So you can actually measure the localizations of 3. So obviously there are going to be far fewer of those in the data sets, but by using those, so it's an independent subset of the data, so independent measurements that actually give us 2-dimensional information, so not just separations but also angles. We could then, again using the Wasserstein metric, compare and contrast the triangles, and essentially using a Bayesian clustering algorithm, group the triangles into a small number of, or into how many distinct sets of triangles there were. We could then compare those triangles, so the separate 2-dimensional information, with our model that came from the 1-dimensional FLImP, and actually identify that they were consistent with what we predicted from the other data. So essentially it was a validation with 2-dimensional information of our model. And so that was brilliant. And what this actually told us, I say we identified a couple of orphan interfaces, and interactions were actually very important. These were actually 2 ectodomain interfaces. No, sorry, ectodomain, sorry, kinase domain, so internal and inside the cell interfaces that were important, and they're actually important in T766M, so particularly EGFR mutation that's important and drives non-small cell lung cancer. So these are actually interfaces that are critical in how that seems to do its work. So we understood, we got an understanding of how that seems to actually how that likes to assemble oligomers and how we might interfere with that. So what was actually done then was a colleague worked with collaborators in London to actually try and interfere with the interfaces we'd identified that seem to be critical to T766M lung cancer. So this is a healthy mouse. This is a mouse with 21. This is 21 days after inoculation, in this case with a T766M lung cancer tumor. This is with a mutation which interferes with one of the 2 interfaces we identified. So this one interferes with an interface that essentially inhibits oligomerization, and we can see that the tumor is much smaller. This is an ectodomain interface, I think, that it interferes with. This last one here is when interfered with, there's a essentially mutant that disrupts an interface which this actually inhibits both oligomerization and phosphorylation, and you can see that essentially inhibiting this interface we identified has almost completely wiped out the tumor. So what this is telling you is that an understanding of which interfaces are important, how they work in this case T760M driven non-small cell lung cancer, was something that we were able to do for the first time using FLImP and employing it along with molecular simulations and lots of brain work. So the potential of disrupting the intracellular interface in non-small lung cancer has been revealed by our new automated FLImP method. Importantly, we'd expect fewer side effects. So this is actually the most powerful one here, is disrupting this kinase domain interface. So that's non-competitive with the normal sort of EGFR binding site, so actually potentially have far fewer side effects. It's obviously harder to do, but this is showing how the detailed understanding at the molecular level and structure function of these signaling complexes that you can now get from these methods is actually potentially opening the door to precise new drug design. So this is work that's currently under review. So there's actually a link here to a preprint. So this is currently we're currently at that point we're waiting for our reviews to come back anytime now. So this has been a huge amount of work, but this is something we could not have done without the automated FLImP and the work we've done with that. So just thinking briefly on what we're going to do next. So we're looking at sort of FLImP operational improvements. So a lot of FLImP we've been developing the method and driving it with our EGFR work. And this has all been done with essentially one Nanoimager running almost 24/7 for several years. We actually do have more Nanoimagers, and so one step is actually to go from operating one 24/7 to all three. And this is where the downside of automation is that you get far more data and you have to do far more analysis, and so you start filling your disks up and you need more computers. So some of the work is just improving some of our efficiency so that we can actually cope with that. But we're now working on, I say, operating all three, so we think we've given ourselves some space to do that in terms of storage and compute. Acquiring more data per slide, so more samples per slide. But we also now are starting to translate our methods to other users. So obviously, we're a national facility, so our methods, we develop methods often driven by a particular interest, but the idea is we can then translate them for others to use those techniques for other problems. So these are our first external users of FLImP who we're working with now to apply it to their problems. But we also have a vision for, and we think there's a lot of room for significant efficiency improvements in terms of both how we data your sample, but also in terms of the compute and storage. We essentially have a first functioning version of a pipeline for which there are lots of areas we know there's room for efficiency improvement. So we anticipate getting significantly faster analysis of the same data and faster results. So making it even easier to do. And as I say, our ultimate vision for the user interface for this is Go. So headless FLImP, so that's something we're interested in, is we don't really, no one needs to interact with the microscope. They put the sample on, click Go, and the next thing you know is when they have these signatures to compare to one another, these FLImP signatures. One more thing, so I mentioned that our improved Markov chain Monte Carlo 1D analysis. So we're particularly interested in objective, robust, and automated, so critically template-free interpretation, structural interpretation from single molecule data. So not just FLImP, there are other techniques, increasingly MinFlux and other super resolution methods that are providing detailed molecular structural information. But a big danger with these is that people can sometimes be tempted to start with what they're expecting, and then you pick that from your data, and then surprise, surprise, that's the result you find. So we're interested in trying to develop methods which take all of the data, ask a well-defined question, and tell you, and give you an objective answer. So that's where particularly using things like Bayesian methods. So we're looking at, for example, extending our Bayesian 1D decomposition techniques to 2 dimensions. To potentially be applied for things like MinFlux. Also can be extended to 3 dimensions. And we're, looking at revisiting the clinical application as a FLImP as a molecular fingerprinting tool, and as potentially as a drug screening and diagnostic tool to, I think, demonstrated the potential it has to impact on the new generation and help discover the new generation of cancer treatments. So in summary, FLImP gives us sort of nanometer resolution molecular separation fingerprints. The automation has now enabled us to increase the throughput and objectivity of our analysis. The Nanoimager was essential to automating this acquisition. It's opened up new avenues to understand lung cancer, and we're looking at applying it further in that direction and along those routes, and also applying it with the user community. And we would like to apply our techniques. We think we'll develop and apply our analysis techniques as well to other techniques in terms of understanding molecular structure from other sort of localization techniques beyond FLImP. And we think we've got room to further increase our efficiency and automation, and and the potential of FLImP as a drug screening diagnostic tool. So thank you for listening. I'm sorry I've gone on for quite a long time. So this is the Octopus team on that's us bonding on an away day recently. This is us looking a bit more calm, and in particular, I say there's a huge number of people been involved in this over the years. So I mentioned Marissa, who's the head of the Octopus facility, and she's also really been the driving force behind the EGFR work. And so the person who sucked me into doing this work in the first place, tempted me in when they needed an astronomer to help analyze their data. So we've got Sarah, who's been working on this right from the start and had been through a lot of the pain of the manual work, and Celine also involved from a long time, and she did some of the mouse work in London. This is Ben, who joined us to do the automation. Michael, who was involved in the FLImP a lot in the past, and Dave, who's also a senior staff who's been involved a lot in helping with this project, and me hiding there. So that's the people in Octopus who mostly currently involved in it. We have a few former members of the group who've moved on now, but who are important with FLImP. We couldn't have done what we've done without the significant scientific computing resources which have been developed over the last few years, particularly scalable service in our sort of data services group and data infrastructure. So people operated the cloud for us and our scalable storage systems. So clinical collaborators in King's College London, Peter Parker, George Santis, and Gilbert in particular, working with Celine on the mouse work recently, but collaborated over many years. And then our amazing collaborators in Geneva, Yanis Galdaras and Francesco Gervasio, who did the molecular dynamics simulations in the paper that's under review. And that would be critical, amazing work there, and funding from a wide variety of bodies, both in terms of the research we do and also for our compute infrastructure. And also just like to thank ONI for inviting me to come and tell you about this. So finally, just one more slide from me. Oh, sorry, if I don't go too far. So we have a few job opportunities in Octopus right now. So there's one that closes this weekend, which is actually for a scientific software engineer, data scientific algorithm developer person to join my team, working on developing and supporting imaging analysis and management workflows, and that's a permanent position. It closes this weekend. We have another software engineer position to join us for 2 years, developing and supporting correlative light and electron microscopy, correlative light and electron microscopy data processing pipelines. And we also have a couple of posts coming up for 2 years on electron microscopy for cryoclem and developing some cryominflux microscopes. So if you're interested in any of those, please, we've had some problem with our job application system today, so some links have gone wrong, but if you're interested, please drop me an email, and we'd love to hear from you. So thank you very much, and thank you for listening, and I think we can move into the Q&A now.
Great. Thanks so much for the presentation, Dan, and I'm sure everyone learned lots of new insights on single molecule imaging and tips and tricks today, so thanks again for that. We're now going to open the floor to questions for Dan, which he will be answering live. And once again, if you have questions, just feel free to drop them in the chat box. And we do have a lot of them already submitted. There's 8 minutes left to the webinar, so we're going to go in full steam ahead, Dan. We'll start with the first one. How important was automation to this project?
So I think, I've probably given a bit of a flavour of that, but yes, I mean, it was absolutely critical. So, I mean, we first developed FLImP, our very first sort of demo of it was about 2011, so it's been going for some time. But as I say, it was very, very painful to do the work. We got some important results, but it was painful, and we were struggling to see how we could keep doing it or offer it to others. So automation was essential to make it something that we could both apply for what interested us most and make available to users. So both the acquisition and the automation, so the analysis automation was absolutely critical to getting this out to users. So yes, basically, the results I just told you about, particularly on the non-small cell lung cancer, would not have happened. I mean, I think I said something like a sort of 20-fold, perhaps to 50-fold, depending on which aspects increase in efficiency. We would still be several years, of full-time, painful, unsustainable work by certain colleagues to have done that without the automation. So yes, absolutely critical.
Great. Our next question: could you have used any other microscope to automate FLImP?
So I mean, we could have used any single molecule TIRF microscope that is fully scriptable and automatable. And I think, as far as I know, that means only is the Nanoimager is the only one. So we identified that. That's what we needed it for in the first place. So we're not aware of any other. So if it, yes, it was. I mean, the alternative would be to automate our own, and we'd probably still be doing that now, or trying to automate the microscope. So so no, we're not aware of any other instrument available that enabled us to focus on telling it what to do rather than how to tell it what to do. So so yes, I don't think we could have done this, certainly not anywhere near as well, if at all, without the Nanoimager.
Great. What has Bayesian analysis brought to this work?
So Bayesian analysis. So yes, I mean we've applied Bayesian analysis in lots of different areas of this. So I mean, for me, I mean, coming from astronomy, I've always been trying to be as sort of objective as quantitative, and that's why to bring those approaches to sort of the sort of bioimaging work that I've been enjoying. So trying to be explicit about our assumptions, for example. So as I mentioned, in terms of ways of sort of techniques for understanding molecular structure from many measurements, if you've got lots of uncertainties in your data, you've got spurious measurements, for example, being as objective and open about what assumption you're making about those, so that you can ask questions and have a reasonable idea of how robust it is and how optimal it is, has been critical. So basically we've tried to apply it throughout. I mean, the single molecule feature detection and tracking is actually using a Bayesian segmentation model. I mean, that's some quite old code now. That's some of the first things I wrote that we applied within here, but that was actually a technique from astronomy for identifying galaxies in images. That there was an early collaboration that actually brought me into the project. But then the more recent stuff, for example, in the decomposition, again, I think it was important in that to actually try and be honest with ourselves about what we can and can't say about our data. So I sort of always try and be as Bayesian as possible in how I formulate things.
Great. All right. Our next question. What motivated your choice of fluorophore? Did you observe any differences with other fluorophores?
So this is I know we've used various different fluorophores over the years. I mean, I've got other colleagues who could probably give a much better answer than me on this, but it's about optimizing them to give us in terms of the photophysics, sort of obviously as bright as possible. What we wanted was fluorophores that we wanted that our photobleaching steps, the actual time between the steps, to be long enough that we could clearly see and we could clearly resolve temporally and have multiple time steps within each level, because that actually helps with our analysis methods. But it was essentially, yeah, I can't remember the whole list, but of properties. I mean, that's something that my colleague Sarah would probably be screaming at me now, saying it was this and that. But we tried various ones, and there are other fluorophores we've used and we can use, but I think we found CF640R. I mean, one thing that's important is trying to have fluorophores that are as small as possible and the linkers that are as small as possible, so that the separations you're measuring as well is what you're measuring is as close to the structure of the molecule that you're labeling, which is also important. But it was basically about how long they last before they bleach, and how bright they are while they're fluorescing, I think was important. And also in this case, I'm not sure exactly, possibly the fact that we wanted to label in the host at the same time, we need to make sure obviously that we don't have overlapping channels so that we can actually do that. So, but yes, I think certain other colleagues could give better answers than me on that one.
Okay, next question. How close are you from being able to implement this in the clinic?
So there's still, I mean, essentially that part of the project, so we made some progress, but as I say, I mean, that was the part of our project that was basically totally disrupted by the pandemic. So I say the collaborators were respiratory, were sort of in respiratory medicine, so they were busy for the last 3 years, and we're looking at how we can re-establish that. But and postdoc essentially wasn't able to do her work for most of the time of her time on that project. But we did make progress. We made some progress to actually in terms of how to label the relevant sample. So it was, as far as we got, it was looking promising. But we basically have to look at currently now how we can revisit that and resurrect that side of the project. So yes, the automation of the acquisition analysis, I say we managed completely, but that was disrupted. So
okay,
we believe it can be done. We essentially, as far as we got, it was all looking very promising. It's just sort of paused as a result of the pandemic.
Sure. Okay, I think we can take one more question, Daniel. We do have a few more that came in, but we do just have time for one more. I may have missed this, but what's the margin of error in localization precision in your experiments, and how do you assess this?
So, I think, as I mentioned, in the actual FLImP fit, so essentially we fit our photo bleaching steps, and we actually use bootstrap resampling. So we essentially, you can imagine the region of interest through time as being, you can unravel it into a set of position and intensity as a function of, well, intensity as a function of position and time, and you can bootstrap resample which of those you include, and you can repeatedly do that. So you can essentially produce lots of bootstrap resample data, each with different randomly chosen subsets of those intensity as a function of position and time points, and repeat your fit, and that actually essentially explores the noise properties of your data without having to model it explicitly. And that gave us our probability distribution for separation for each measurement. So it gave us the curve where the peak, and so essentially we could get a mean and standard deviation, for example, or take other measurements. So essentially we use bootstrap resampling of our region of interest through time to empirically understand, and we demonstrated in early papers how this is reliable. And it means when you switch detectors, you don't have to come up with a new noise model. It empirically does it every time. And so that's how we do it. I say in the early days we were getting perhaps, so we don't really, I mean, we can get it, we don't really quote it in terms of the localization area of the molecules. We look at it in terms of the uncertainty of the separation measurements, which went down from about typically perhaps 5-6 nanometers in the early days to, in some of our more recent data, we're getting data with 2 nanometer uncertainty or confidence intervals, essentially sort of 70% confidence intervals in our separation measurements. So yeah, we're sort of nanometer sort of scale localization.
Great. All right, and it seems as if we are now at time. So a very big thank you for the presentation again, Dan. If we were unable to get to your questions, we will forward them to Dan, and he will do his absolute best to answer them individually. But once again, this webinar was recorded. Therefore, if you need to refer back to it, you can. And Dan mentioned a few different tips and tricks webinars that we hosted in the past. So all these recordings can be found within the resource section of our website. So please just make sure to check that out. And thank you to all the attendees for joining us today. And upon exiting the webinar, you'll receive a survey to fill out just to give us overall feedback on your experience today. And thank you again, and we hope everyone has a wonderful rest of the day.
Thank you.